Hi, it's Mr. Bennett. In this video, what we're going to talk about is we're going to talk about genetic engineering. Now, when you hear the term genetic engineering, you may think of some crazy stuff like these glow-in-the-dark cats. This is not Photoshop. This is an actual thing where we created cats that have jellyfish DNA in them, so they make a protein in their hair that makes them glow in the dark. So this is probably the first thing you think about when you think about genetic engineering. But genetic engineering isn't just taking genes from one animal and moving them to another. Genetic engineering, what it is it's anything humans have done to modify the genes of an organism. And we've been doing this for a really long time. That picture there, that's of the ruby red grapefruit. And we created the ruby red grapefruit way back in the 1950s. Now, we didn't create this grapefruit by taking a gene from one organism and putting it into a grapefruit. We actually made it by sheer chance, trying to make the plant mutate until eventually we got the color we want. And so genetic engineering has been around for a long time. Now, there are several different ways you can go about doing this. So genetic engineering is any time you modify the genes of an organism. Now, sometimes it's going to involve in the lab where you're taking DNA from one species and putting it into another. Other times it can involve far more simple methods, like, for instance, selective breeding. Selective breeding is an example of genetic engineering. That 12,000 years ago, every dog on the planet was a wolf. There was no other type of dog on the planet except for the wolf, really. And all these other different breeds of dog that we've created have been the result of selective breeding. Now, the end result of that is we've changed the genes because, for instance, these dogs down here obviously have different genes from a wolf. How did that come about? Because of humans. We controlled who got to mate together, and so we controlled the genes, and we changed the genes of an organism. Other methods you got, inbreeding. We'll talk about that. Kind of a scary topic, but that's another way that we modify the genes of an organism is through inbreeding. We're also going to talk about transgenic animals. An example of that is going to be our cats over here. Transgenic means you're taking a gene from one species and putting it into another one. And finally, the last example of genetic engineering we're going to talk about is we're going to talk about cloning. So the first type of genetic engineering that we're going to talk about is we're going to talk about selective breeding. We're going to talk about this first because this is what humans have been doing the longest. Humans have been doing selective breeding for over 12,000 years. And what selective breeding is humans control what organisms can mate together. And the ones that we let mate together are the ones that have the traits that we want. Let's look at this top picture here talking about the chickens first off, okay? This is what a chicken looked like in 1957, okay? They weighed about 900 grams. Okay, that's how much they weighed. By 1978, you can see they got bigger, and then by 2005, this is what a modern day chicken looks like today. These suckers weigh 4,200 grams. And so what's happened is we've increased the weight of the chicken by over 6 times in just 60 years. Now, how did we do that? We only let the largest chickens mate together. And so if you're a skinny chicken like today, a chicken like this would never be allowed to mate because we don't want a chicken like that. We want this guy, okay, that's got these large breasts on there that are just delicious to eat, and off we go from there. And so this guy today would not be allowed to breed, and we would only allow the chickens that have the largest size to mate. And if you carry that out for 50 years, you can see the progression. By only letting the largest animals mate for 50 years, we've eventually made a much, much, much larger chicken. Another example of selective breeding is apples. This right here is what wild apples look like, okay? They're not very tasty. They're pretty small. They're about the size of a golf ball. That's all the bigger they are, okay? They're very crunchy. The size, not the greatest, and the taste isn't the greatest. Now, what's happened is humans have slowly, through selective breeding, created all these other different types of apples. How did we make all these different types of apples? By controlling which apple trees we let mate with each other. By only allowing apple trees that had desirable traits mate with each other, we were slowly able to turn wild apples into something you actually want to eat. Think about it. Average wild apples the size of a golf ball. Now you've got them the size of a softball. How did we make that happen? Same way we made the bigger chickens. We only let certain apple trees cross with each other, and the only ones we let cross with each other are the ones that had the traits that we wanted. Now, sometimes when you go about the process of selective breeding, you're going to do something that we call hybridization. And what hybridization is this is where we cross organisms with different traits to get the best of both worlds. Corn is a great example of this. That if you talk to anyone who remembers corn back in the 1930s, we didn't have these giant ears of corn. Your options were you could have a big ear of corn like this, But as you can see, all the kernels do not completely cover the ear of corn, or you could have a small one like this guy where the kernels completely cover the ear of corn, but he's maybe 7 inches in length if you're lucky. And so what they did is they crossed these two together. They crossed the varieties of corns that made big ears with ones that had small ears, and what they were hoping to do was they were hoping to make a variety of corn that would make big ears, and so it would have the trait of this parent, and then it would fill in that ear completely, so it would have the trait of that parent. And that's exactly what happened. So that's hybridization. Hybridization is when you cross organisms with different traits and you're trying to get the best of both. So an example of hybridization is the Labradoodle. The Labradoodle is a breed of dog that's made by crossing a poodle with a lab. Poodles are the third smartest breed of dog on the planet, but they're not big enough to really do a lot of good. And so the idea was they crossed them with labs to make a bigger dog that was smart, and that's the Labradoodle for you. Now, when we do hybridization, we make hybrids. And what a hybrid is, it's more than just a car. A hybrid is an organism that comes from crossing different varieties or species. And here's an example of a hybrid for you. This is called a pluot. And what a pluot is a pluot is a cross between a plum and an apricot. Okay? So this is a hybrid species. It's got its parents are two different species that we're able to cross together. And the result is a fruit that's kind of halfway in between. Okay, it's going to have some characteristics of plums, but check it out. We got a pit going on there, kind of like an apricot. So that's selective breeding, and this is the oldest tool humans have used, but the end result of selective breeding is we have changed the gene composition of these different species. That obviously this species of corn right here is going to have much different genes than either of these guys, and obviously our big old chicken over here is going to have different genes than the ones that came before him. This is a result of humans engineering these animals to be what we want them to be. Our next example of genetic engineering is going to be inbreeding. Now what inbreeding is where we breed organisms together that have very similar traits or very similar characteristics, or they're the same breed. Okay, so an example here, what you're looking at, this is a purebred border collie. Purebred border collie. So what that means is both of his parents were also a border collie. Now why would we want to do this? Well, because this is the smartest dog in the world. And you don't want to mix him with something else. You mix this breed with any other breed of dog, you're going to make him dumber. And so that's why we breed border collies together, is we've isolated exactly the traits that we want. We've got the smartest dog in the world, and if we breed it with anything else, we're going to make it dumber. And so inbreeding is what you do when you have all the traits that you want and you want to save them. Because if you mix it with something else that has different traits, you may lose those beneficial traits. And so that's where inbreeding comes about. Now, you see this term here, purebred, okay? It's a term you're probably familiar with, but the definition of a purebred is these are organisms whose parents are of the same breed or the same variety. So I've already talked about how this guy up here, this dog, is a purebred border collie. Both of his parents were border collies. Here down here, that is a purebred Holstein cow. Both of its parents were Holsteins. And so he's going to have all the same traits of a Holstein. They're not going to be lost. Now, there's some definite benefits to inbreeding. As I've already mentioned, is that good traits are more likely to be passed on. That purebred animals tend to be more homozygous because their parents were the same. As a result of that, if you breed two species together, two individuals together, they're both homozygous for the same trait, they're obviously going to pass that trait on to their kids. And so if you're talking about cow and Holsteins, the big trait of Holsteins is Holsteins give more milk than any other species. These guys are the champion milk producers. And so that is a trait we want to maintain. And so that's why we breed Holstein cows together, to make sure that we make animals that maintain that trait of giving lots and lots of milk. Now, the downside of inbreeding is homozygous is a two-edged sword. That sometimes homozygous is a good thing if it's a good trait, but remember, a lot of our bad traits are recessive. And if you are more homozygous, you're more likely to show that bad trait. So you may save the good trait, but you may also make it so that bad traits show up. This graph up here, what we're looking at, this is showing what percent the Holstein breed in America has become inbred. And if you look at the graph right here, your average Holstein cow in America is approximately 8% inbred. So it's 8% related to itself. because what we've done is we've continually bred Holsteins together and we haven't brought in other breeds. The cows have become more and more inbred. Well, the problem with that is we're seeing some nasty things show up as a result as the cows become more and more inbred. What you're looking at down here is calving interval. The more frequently a cow has a calf, the more money she's going to make you. And you can see in the year 1987, our calving interval for cows was 12.5 months. That every cow in America, on average, was having a calf every 12.5 months, or roughly every year. By the year 2003, that number's gone up to 13.5, and today that number's almost 14. And so by inbreeding cows to make it so that they make more milk, we've also made it so they're having less babies, and so that's a downside. A more extreme example of the downsides of inbreeding is, check out this guy right here. Now, this guy is a white tiger, and white tigers are extraordinarily rare. It's a recessive gene that isn't out there very often. But thanks to Siegfried and Roy and everything, white tigers is something that everybody wanted. Every zoo wanted to get a white tiger. So to make more of them, what they did is they did some pretty severe inbreeding of existing white tigers to make more. And all of a sudden we start having bad things show up. This white tiger right here, his dad and his mom were brother and sister. So we crossed brother and sister together. They were both white tigers, and we made this guy. The reason why they did that is so that they could guarantee that they got a white tiger out of the deal. Well, you can see that he's got some obvious issues with him. He has all sorts of deformities and so forth that make it so he's probably going to have a very short life expectancy. And this is the downside of inbreeding. So the upside of inbreeding is you're going to become more homozygous, which makes you more likely to keep the good traits that you developed. The downside is homozygous is also how bad traits show up, which could end up killing you in the long run. Our next example of genetic engineering is the one that most people think of when they think about genetic engineering, and that's going to be transgenic organisms, more commonly known today as GMOs, or genetically modified organisms. And what a transgenic organism is when we take genes from one species and we put it into another. Now, we got a couple of ways we can go about doing that, but to put it down real simple, we have transgenic animals, we have transgenic plants, we have transgenic bacteria. In animals, what we do is we take the DNA that has the gene that we want to put into our animal, we isolate that DNA, and then we inject that DNA into a fertilized egg. It needs to go into a fertilized egg because we need to make sure that every cell in that animal has that gene. Well, the only way you're going to make sure you do that is if you start out with the very first cell that animal starts out with, and that's going to be a fertilized egg. So we put this gene, we inject it into our fertilized egg, and then we take our fertilized egg and we drop that into a mother to be a surrogate, and then however long that animal is pregnant for later, we end up having our baby, and that baby is going to have the DNA that we injected into it. So that's the basic process of how you make a transgenic animal, is you've got to take and find the gene, put it into the fertilized egg, raise the thing up, and now you have your transgenic animal. Now, if you take and you mate this guy with another transgenic animal, he is going to pass on that DNA to its own kids. Because that DNA, because we put it into the original egg, every cell in that animal's body is going to have it, including its gametes. Now, we've been doing transgenic stuff for a long time. First examples was the late 70s. I've talked about this when we used bacteria to make the protein insulin. And how we went about doing that is we took the DNA out of humans that has the instructions for how to make insulin, we put it into bacteria, and now we have bacteria that make insulin for us. And so all processes of genetic modification or transgenic modification involve taking DNA from one species, that's the DNA instructions for how to make the trait that you want, and you go ahead and you slap that into another species. So why do we want to go to all the trouble of taking DNA from one species and putting it into another? Well, there can be some major benefits by doing this, where you can make existing animals be more beneficial to us. One of the first examples of a transgenic or genetically modified organism that was made for humans is this. What you're looking at here is over here, this is called golden rice. Okay? And you can see the reason for it. Okay? It has a definite yellow tint for it. The reason why we make golden rice is normal rice lacks vitamin A. It is very, very, very low in vitamin A. And if you don't get enough vitamin A in your diet, you go blind. And so what was happening is lots of people in India and Asia and Africa rice is the majority of their diet. And because they were mainly eating rice, they were getting very low levels of vitamin A, and they were going blind as a result. And so what they did with golden rice is they actually took a piece of DNA from from a plant that makes lots of vitamin A, and that would be the carrot. The reason why carrots are orange is because they make a compound called carotenoids, and carotenoids are used to make vitamin A. And so they found the DNA in a carrot that makes carotenoids, and they went ahead and they put that DNA into the rice. And so what you got going on here is this rice over here is making a carrot protein. And that carrot protein that it's making is making it so it's super high in vitamin A. And because it's super high in vitamin A, when people grow this golden rice and they eat it, they don't go blind. And so here's an example of why we want to do genetically modified stuff, is it's preventing people from going blind. Other example of a GMO crop is corn. What you're looking at here is we've modified corn to make a protein that we call the Bt protein. And what this Bt protein is it's a protein that's made by bacteria, and it kills caterpillars, and only caterpillars. That's the thing that's really cool about it, is it won't hurt any other insect, it won't hurt humans, it won't hurt any other plant. What you got going on here is you can see this corn here is being eaten by caterpillars. This corn up top isn't touched by it. That's because this corn up top has a piece of DNA from a bacteria, so it makes this Bt protein, which kills the caterpillars. Now, the reason why this is a good thing is this corn does not have to be sprayed with pesticides. We know that pesticides aren't great for people, and the less pesticides we can use, the better it will be. And so genetically modified crops are used to make it so that we can use less of these dangerous pesticides and not have all this food spoilage, because we're using natural proteins from bacteria and so forth that will kill the insects for us instead of pesticides. A couple other examples of transgenic organisms: cows. Okay, we've got transgenic cows out there that we've modified cows so that in their milk they actually produce proteins that can be used to treat people for diseases. And so that's an example of transgenic organisms, that we've identified proteins that can treat people, and an easy way to get people lots of these proteins is just throw it into a cow, and then cow, when it makes the milk, is going to make the proteins. Or the other example you've heard me use a lot is that spider silk is extraordinarily useful. It's 10 times stronger than steel. The best bulletproof vests on the planet are made out of spider silk, and it's used for all sorts of other things. But the problem is, no one wants to keep enough spiders to make enough silk to make a bulletproof vest. And so what they did is they found a piece of DNA in a spider that's the code for how to make the protein of spider silk. They went ahead and they put that DNA into a goat. And then what happens is when they milk these goats, their milk contains spider protein. They can isolate that spider silk protein, and use it to make bulletproof vests and parachutes and everything else. And the goats can make this protein a whole heck of a lot cheaper than you can afford to raise the spiders to make it. And so those are just a few of the examples of transgenic organisms. We use it to make plants so they're more drought resistant, so they grow better, so we don't need to use herbicides, all sorts of things that we use GMOs for. Are they safe? You betcha. You betcha. That a lot of people freak out about GMOs, that, oh, we're putting foreign DNA into these animals and that DNA is going to take us apart, is going to take us over. No. That, yes, this rice over here has a different piece of DNA in it, but when you eat something, DNA is all the same. Your body breaks DNA apart so it doesn't work anymore. So it doesn't matter where the DNA came from. Any DNA that you eat, your body's going to break apart, and it's not going to be a big deal. Our last example of genetic engineering we're going to talk about is cloning. Now, what cloning is basically is you're making an organism's identical twin, but they're not going to be born at the same time. What we do is in cloning is we can make an organism that's genetically identical to the original. So cloning would be to make an exact copy of you, another human that has all the exact same DNA that you have in you. Okay? For the longest time, we thought this was impossible, that we've cloned bacteria before, but we didn't think we could do it with mammals until the year 1997. In 1997, everything changed, and it changed with Dolly the sheep. And here's Dolly right over here. Okay? And here's her baby down here. I think the name is Molly. Now, Dolly was a clone of another sheep that was 4 years older than Dolly. That what they did is they took a cell from the udder of a sheep, all right? They took the DNA of that, they took the DNA out of that cell, and then they used that to clone Dolly. How did they do that? Well, they took a second cell that was an egg cell and they removed the nucleus from that egg cell. And in its place, we put the nucleus of this cell that we got from the donor animal. So now what we got is we got an egg cell over here that has the exact same DNA as the donor animal. We then took that egg cell, we got it to divide a little bit, we implanted that into a surrogate mother, and 120 days later, Dolly was born. So that's how the process of cloning works, is you take the DNA all out of one cell, put it into an egg, make that egg think it's a fertilized cell, you have a clone. While we're here, why not? The reason why she's called Dolly is the cell that they used to clone Dolly came from the udder, which is another name for mammary tissue, and so they named her Dolly after Dolly Parton. I'll let you figure out the joke from there. Now, since we've cloned Dolly in the year 1997, we've cloned lots of stuff. We've cloned cats, deers, dog, horse, mule, ox, rabbit, rat. Actually, the team that cloned the mule for the first time, one of the professors is actually at Utah State. So we went on a big thing with cloning there for a while. And there can be some big advantages with cloning. There's a big factory in China right now where they're cloning cattle like crazy because they think it's going to be a cheaper way to make cattle. Now the question comes up, what about humans? Could we clone a human? You bet we could. Have we? No. The reason why we haven't is because there's very severe laws against cloning humans, that we're okay with cloning a sheep. It's just a sheep, whoop-de-frickin-do. But the idea of cloning a human makes humans kind of antsy, and we're not sure it's something we want to do. Is the technology there to do it? Sure. Have we done it yet? Not yet. Are you probably going to see it in your lifetime? Wouldn't surprise me in the slightest. And so that's cloning for you, and that's another example of genetic engineering.